Plasmids. Who doesn't like a good circular piece of DNA that replicates outside the chromosome, is incredibly versatile as a genetic scaffold, can move around and can confer antibiotic resistance. And even though resistance to our own antibiotics sounds like bad news, they are critical genetic identifiers, so we can observe and manipulate these small pieces of genetic material to do our bidding. So why would we bother trying to remove these handy genetic elements, you may ask? Well, it's worth the effort if you want to create background strains, ones that don't have native plasmids.
And through these plasmid-free bacteria, we would be able to develop safe industrial-grade microbes that don't harbour any residual antibiotic resistance, creating better food products. It must also be said that plasmids can carry genes that are only temporarily needed by geneticists, meaning that they actually have a higher chance of potentially hindering genetic engineering, rather than aiding it. Plus, background strains allow us to study the functions of genes on plasmids, allowing us to properly understand the role that bacteria play, either in pathogenesis or as normal flora. This is because plasmids contain whole gene clusters that can control things like virulence factors and metabolism, and so much more. The possibilities really are quite endless.
The problem is, there really aren't many ways to cure plasmids. The staples are either agent-based curing or plasmid displacement, both of which are really, really inefficient. And the former, agent-based curing, involves subjecting many bacterial generations to physical and chemical stresses, so they expel the plasmid, or can involve placing them into a non-stressful environment so they can expel the metabolically cumbersome plasmid so they can replicate faster. Whilst the latter uses transformation with an unstable and incompatible plasmid, which then interferes with the resident plasmid, causing it to be lost. Both of these methods involve a lot of time and effort, and all the while running the risk of accumulating unwanted mutations in the bacterial genome itself.
So essentially, it looked as though the plasmid curing methodology had bottlenecked, until it came to Crispr, or clustered regularly interspaced short palindromic repeats, was discovered. This versatile genome editing tool was discovered as a sort of bacterial immune system, in which it cleaves and digests invading viral or plasmid DNA, using the Cas9 nuclease. To begin its function, the nuclease is led by a short 20 nucleotide long guide RNA, or gRNA, and that can bind to the beginning of any DNA sequence. This self-defense enzyme can then be exploited to recognize any sequence that we desire and start cleaving and digesting at that site.
When speaking about how the CRISPR-Cas9 system would really speed up the research process in microbiology, Mycobacterium tuberculosis comes to mind. This mycobacteria, as its name suggests, causes tuberculosis and results in 1.5 million deaths worldwide annually. And as a result, a lot of research has been poured into this bacterium. But we've only scratched the surface. For instance, even though we know that the plasmids in the tuberculosis-causing bacteria are involved in its pathogenesis, we do not understand how or why the plasmids contribute to disease.
So the two main problems faced when studying this bacterium are the fact that it's very slow growing, as well as that it doesn't take up DNA very efficiently. So if we relied on the old and unreliable methods of plasmid curing, growing a plasmid-free strain seems almost impossible. However, using the CRISPR-Cas9 system, this could create plasmid-free strains of M. tuberculosis much more quickly and efficiently. This rapid production of plasmid-free strains would contribute heavily to the increased ability to decode and decipher the plasmid's contribution to the bacteria's virulence.
On a more health-positive note, plasmids also play a major role in normal gut microflora, like the lactobacillus species. These great bacteria are used extensively in probiotics due to their association with good gut health. 38% of these bacterial species within this genus utilize plasmids for regulating antibiotic and metal ion resistance and reducing bacteriocins and bacterial adherence.
Similarly, as described in the previous case with tuberculosis, creating background plasmid-free strains would be super useful. Developing these mutant populations would be the starting point in categorizing the role that these plasmids play in the benefits of these bacteria in probiotics and in the human gut. On top of that, we could finally settle the pseudoscience involved around probiotics and potentially their disease applications.
If we now take a look at the newer advances in science, Lauritsen et al. Developed a universal one-step plasmid curing system named P3. They created a plasmid with multiple guide RNAs and one Cas9 gene, and then transformed the target bacteria with the created plasmid. The trick is that the P3 plasmid actually contains 4 guide RNAs, meaning that there is a high chance that all, or at least most bacteria would contain the complementary sequence to at least one of the guide RNAs. And hence the claim that the P3 plasmid would be universal. However, it was found that the curing efficiencies were actually between 40% and 100%. So suddenly the universal claim doesn't quite ring true anymore.
However, their claim to being a one-step process does ring true. As seen on the figure, the only thing that's required is that the bacterial cells take up the plasmid. And this is only one step. However, this is not the best part. The P3 plasmid is not only efficient, it can cure multiple plasmids in the same cell in under 24 hours. And remember how we said plasmids can potentially hinder genetic engineering? Well, the P3 plasmid is able to self-cure, meaning that it removes itself after performing the action. So maybe the 40% curing efficiency is maybe just the plasmid being a little bit too efficient.
Not only does CRISPR-Cas9 revolutionize genetic protocols in the lab, but it may also have important clinical applications. Antibiotic resistance in bacteria is a scary prospect. For example, in the early 1980s, labs developed a new broad-spectrum beta-lactam antibiotic, but within a few years, enzymatic resistance to those antibiotics had already been discovered. And the worst part is that the plasmids are actually responsible for the resistance, so it could be horizontally transferred to other bacteria. And if we are able to find a way to efficiently resensitize resistant bacteria to antibiotics, it would be a Nobel Prize-worthy feat.
So this method of resensitizing bacteria to antibiotics was explored by Kim et al. So they did a similar project to the previous study by Lauritsen, and essentially they introduced a plasmid containing the Cas9 gene and gRNA into resistant E. coli cells, which took up the plasmid and were transformed. After overnight culturing, the bacteria were then plated out, and they found that 99% of these bacteria were now sensitive to the antibiotics which were in the plate that they grew on. So this showed that the detrimental transformation of the plasmid containing Cas9 nuclease and gRNA was expressed by the bacteria, and that these antibiotic-resistant plasmids were then cleaved by this introduced system and were unable to rejoin the cut ends, leading to their removal from the cells and them regaining sensitivity to the antibiotics.
So it turns out we might actually have a saving grace after all. But this is only the in vitro side of the story. Who knows what it would be like outside the lab? And plus, we have one major issue to address, where each antibiotic resistance plasmid has different DNA sequences that confer the same or similar effect. This means that one guide RNA will not do the job. Therefore, it is vital that we find a universal sequence that actually lets us exploit to remove the protections the bacteria has to our own medical arsenal.
With so many current breakthroughs in genetic technologies, it's really an awesome time to be involved in molecular sciences. Manipulation of plasmids through CRISPR-Cas9 cutting has enabled quick and easy plasmid curing of bacteria. This process would revolutionize the understanding of plasmid gene functions, whether in pathogens like M. tuberculosis or in human microflora like lactobacillus. Furthermore, it may even help us combat the ever-increasing rate of antibiotic resistance in bacteria by cutting and removing resistance genes on plasmids. Even though the efficiency and accuracy of the process still needs some improvement and tweaking, we owe most of our important advances in bacterial genetics to our CRISPR-Cas9 system of genomic editing.